Hello everyone, in case you don't know me, my name is Meredith.
And I'm Laura.
And we did our project on Glycerol Ester Hydrolase, more commonly known as triacylglycerol hydrolase. And this is just an image of our enzyme. So before we get started on more detailed information, I want to give you a general background about our enzyme. Our enzyme is classified under two enzyme groups, lipase and hydrolase. Lipase is kind of a subcategory of hydrolase, so we're going to talk about lipases first. Lipases are enzymes that catalyze the hydrolysis of oils and fats. We're going to be focusing more on oils, and in doing this, they create free fatty acids, diglycerides, monoglycerides, and glycerols. Lipases have a high stability in organic substances. They also perform synthetic reactions in organisms. They also have a broad specificity, which we assumed meant that they have an induced fit model. And they also work in milder conditions. This enzyme, lipases, are found in almost all living things, all living organisms, animals, plants, fungi, bacteria. And something special about it is that it contains a mobile subdomain called a lid. This lid has open and closed formations, and the lid helps to regulate substrate binding.
And then the hydrolases. So as you know, hydrolases use water to break up bonds. Specifically, our enzyme is in the alpha-beta hydrolase family, meaning it has a highly conserved catalytic triad. The catalytic triad consists of one nucleophilic residue, typically serine, one catalytic acidic residue, either aspartic acid or glutamic acid, and then one histidine residue. And then here are models of what the lid looks like opening and closing. In the space-filled model, as it opens, you can see it has a very hydrophobic pocket, and that orange part in there is the catalytic triad. And the lid comes into the open conformation when it comes into contact with a lipid-water interface. And then here's the reaction that our enzyme performs. So it is in the enzyme class of 3,1,1,3, meaning that it takes triacylglycerol plus water, which is the hydrolase part, and breaks it into diacylglycerol and dicarboxylate. Diacylglycerol is one of the products formed from a lipase, so that's the lipase part of it. And then in our paper that we chose, they look into African oil beans. So oilseed lipases are actually more active with short free fatty acid chains, similar to this generalized reaction that we have here.
So this is a slide showing the organisms, or some of the organisms that our enzyme is present in. First, we have humans. In humans, it helps to reduce obesity to an extent because it helps to break down fats in the body. In mice without this enzyme, particularly females, are not able to nurse because it reduces milk production. Bacteria are special in this case because they have a cofactor of calcium, and in this diagram you can see the green dot is where the calcium would bind, and this will be significant a little bit later on when we talk about our paper. And speaking of paper, we focused on the African oil bean, the researchers focused on the African oil bean, and so beans are also a category in which this enzyme is present in. We did some research on bioinformatics, and we found that, we looked into the peanut and the castor bean. We looked at both of these because they were both mentioned in the paper that we looked at as references about the African oil bean because they are similar. We have length here, mass, protein, or similar proteins, as well as domains, and in the yellow highlighted you can see the domain. And that was peanuts, and this is castor beans. We have some of the same similar information, and again we chose to do this because these two, both the peanut and the castor bean forms of the enzyme are very similar to the African oil bean enzyme in structure as well as function.
Now for the methods and results. So the actual paper we chose to look at is titled Lipase Activity in Dormant Seeds of the African Oil Bean. This is what an African oil bean actually looks like. So as we've been saying, we chose this paper because it looked at an enzyme in an organism other than humans, because humans have been so widely studied. Also, our paper mentioned that upcoming research has shown that it's possible that seed lipases could be used as substitutes for microbial lipases in bioengineering and organic chemistry and other science fields. The purpose of the paper was to see how varying conditions affected the stability and functionality of our enzyme triacylglycerol hydrolase. The researchers looked at pH, temperature, time, ion, and substrate concentration, enzyme concentration, but along the way they ended up looking at substrate specificity as well.
So before we get into much more detail about the experiment itself, we're going to talk a little bit about the model organism that the researchers used. They looked at ungerminated or dormant seeds, which means that the seed had little or no lipase activity. Specifically, their model organism was the African oil bean, like we've previously mentioned. And this, the African oil bean is a little bit different in that it has lipase activity, but only when the seed is disturbed in some way from its normal conditions. So like extreme temperature, extreme pH, physical contact, things like that will disturb the bean, and that will activate the lipase, which will destroy the inside of the bean. And these are the methods, a list of methods that the researchers took. And we're going to go over all of them in much more detail, but for the first one, picking and storage, I'm going to talk about that a little bit. First, the researchers had to pick the beans, obviously, and then they stored them in 4 degrees Celsius in order to kind of prevent lipolysis.
And then enzyme preparation, they isolated the enzyme by creating acetone powder that was prepared following the processes in this 1986 paper. The actual processes involves cracking the seeds, and they avoid bruising the inside. They obtained 25 grams of the seed cotyledons. They ground and mixed it all together with cold acetone, and then they filtered and washed it with cold acetone 4 times and let it air dry. And then they created an assay to measure lipase activity. The actual assay was made of a mixture of substrate, which was either palm kernel, raffia, or coconut oil, hexane to dissolve the oil, and then the crude enzyme, which was the acetone powder I just mentioned. After an incubation of 1 hour, free fatty acids were obtained. Free fatty acids were quantified with a sodium hydroxide titration, and percent of free fatty acids released expressed lipase activity, which is how the researchers examined the effects that all of the factors that we had mentioned affect the enzyme. So first, some of the first factors, they looked at how pH affected enzyme function, or lipolysis. They used phosphate buffers ranging from a pH of 4 to a pH of 9. They looked at temperature ranging from 30 degrees Celsius to 80 degrees Celsius, and then they also looked at incubation time, where they kept temperature constant and varied incubation time from 15 minutes all the way up to 60 minutes.
And our next factor is ions. The researchers looked at ions, sodium chloride, calcium chloride, mercuric chloride, and EDTA. They used a 1985 study, and they took 1 milliliter of 0.01 molar solutions of each of these ions and added it to the assay solution, and then they allowed it to an incubation period of an hour, and then they looked at the lipase activity. Next, and the last factor that they looked at was enzyme concentration. They varied the lipase concentration from 0.25, 0.5, 0.75, and 1 grams, and they also had a control group that did not have any acetone powder in it, and then they were able to observe the lipase activity.
Now for the results that the researchers found. First, while they were not originally looking at it, they did find some results when it came to substrate concentration and substrates in general. So first, in the graph, you have substrate concentration on the x-axis and rate of lipolysis on the y-axis. Up to 5 grams of substrate concentration, lipolysis increased, but after 5 grams, lipolysis started to decrease. Then they looked at substrate type. So for coconut oil and palm kernel, lipolysis was highest because those two oils are both short-chain free fatty acids, which I had mentioned earlier. Oil lipase, seed oil lipases work best with short-chain free fatty acids.
And the next results that they found were involved with pH, temperature, and time. The first one, pH, they found the optimum enzyme activity at a neutral pH. For temperature, they found the highest activity at a temperature of 30 degrees Celsius. And as you can see here, for time, they found the optimum lipase activity at 60 minutes. And for ions, so for the ions they looked at were the sodium chloride, calcium chloride, mercuric chloride, and EDTA. For calcium and calcium chloride, they found that it increased lipase activity by 64%, which is pretty significant, and this allows us to predict that our enzyme may have a predisposition for calcium and working well with calcium. And this is supported by the fact that I talked about earlier with the bacterial form of the enzyme having a cofactor of calcium. And in our research, we also found a similar enzyme, which is pancreatic lipase in humans, and this also has a calcium cofactor as well. The other ions, which were mercuric chloride, sodium chloride, and EDTA, all inhibited the activity of lipase. Sodium chloride with 36% inhibition, mercuric chloride with 28.5% inhibition, and EDTA with 28% inhibition. And the researchers also pointed out for mercuric chloride that this inhibition might have been due to a sulfhydryl group.
And then now the results for the enzyme concentration. Up to 0.25 grams of enzyme concentration with substrate concentration being kept constant at 5 grams, there is a steady increase of lipolysis. And then between 0.25 grams and 0.75 grams of enzyme, lipolysis greatly increased, and after 0.75 grams of enzyme, lipolysis started to drop off. So this shows that enzyme lipolysis was highest at 0.75 grams of enzyme. And then just to wrap up everything we've been saying, so for the results, our enzyme works best at 30 degrees Celsius, a neutral pH, and 60-minute incubation time. Calcium ions increased or enhanced enzyme activity while all the others inhibited it. And then in general, our enzyme glycerol ester hydrolase did follow the conditions that lipases generally do, that they work best in mild conditions like the room temperature and the neutral pH. And they also perform the same processes like the reaction that I showed, and then the lid and all that stuff. So that's the end, and then here's our bibliography.
Thank you for watching.